So we kind of got a good glance into the enzymes, right? Now, before we move on, let's do a quick, dirty summary of what we've learned so far. First of all, 3 major nutrients that provide building blocks, chemical building blocks and shit. I mean, three major nutrients that are sources of energy, chemical building blocks: carbohydrates, proteins, and lipids, right? Got it? Okay. Now, they are broken down. In the mouth, which 2 are broken down? Carbs and fats. In the stomach, fats and proteins, yes. And in the duodenum, all 3, right? We vaguely remember that enzymes that break them down, which is fine. Now, one important thing about breakdown in the duodenum: those molecules, the proteins, the huge carbohydrates such as starch, they're big, right? Like, they're really big. Like, protein that contains 100 amino acids is a small protein. We talk about 400 amino acids as a good, decent sized protein. Collagen contains, I don't know how many. I think individual molecule can mount up to 500. No, 50,000 amino acids. I mean, collagen is gigantic. It's huge. It's ginormous protein. Okay, starch is huge. Okay, triglycerides are slightly smaller. Does that make sense? So, enzymes that are in the pancreatic juice, what they do? They take what? A big molecule and break it down in a smaller chunks, which is still remarkable. Okay? Now, brush border enzymes in general take those smaller chunks and break them down into individual building blocks like amino acids or monosaccharides. Okay? We got this. I just want this process without details yet to get into your head. Okay? So you have this idea, in the outskirts, big chunks are produced immediately at the cell membrane. The units are made. Let's take a look at the amino acids, proteins, essentially. Okay. Now, proteins are big. You can see them right here. Okay. That makes sense? Now, these proteins get broken down into the smaller chunks. Let's say this is a smaller chunk. Okay. Different pancreatic proteases will break them down. They will also, of course, produce a bunch of individual amino acids together with the brush border enzymes. So far, does that make sense? Okay. So what you end up with is the bunch of amino acids that are sitting right next to the apical membrane. Okay, apical membrane of the columnar cells of intestinal epithelium. Can amino acids diffuse directly through the membrane? No. Because they are what? Water soluble. Right? So how can we bring them in? Through some sort of a transport molecule. Okay? The next question is, we didn't talk about it yet, but try to think. Amino acid concentration, is it going to be high in the cell or low in the cell? At pretty much any point. It's kind of a generalized question. Generally high. Because, yeah, cell has to maintain very high concentration of amino acids because cells constantly produce proteins. Okay? So, even if we provide them some sort of a transport protein, can they go in by diffusion? No, they will have to go against the gradient. So we have to actively transport them. For this, we have a mechanism of the secondary active transport. Okay? Does that make sense? And that extremely beautiful rendition of the mechanism underlying secondary active transport you can see in the right bottom corner of the slide. The one, does anyone remember what is the secondary active transport? Can anybody explain it to me in the simple terms? I don't need the sophisticated explanation, just what's going on during secondary active transport. Yes, that would suffice. For now, that would suffice. Something else also goes into the cell. So that something else actually provides the energy for the transport of molecule of interest. So, molecule of interest in this case is what? That we need to transport against the gradient. Amino acid. I don't like where I positioned this. So let me try and position it differently. So we're going to make it red. Oh, come on. So that's gradient, and that little red thing is amino acid, right? And it has to go up the slide. In order for it to go up the slide, like if you want something to roll up the slide, what do you have to do? Push it up. How can we push it up? We have to take something that rolls from another slide so it can push the red ball up. So in this case, red ball is amino acid and the green ball is sodium ion. So here you see sodium diffuses into the cell from the lumen. From the lumen of intestine, sodium diffuses into the cell. Does that make sense? It diffuses because sodium concentration in the cell is very, very low. If it diffuses and diffuses and diffuses, what's going to happen to the sodium concentration in the cell? It's going to go up. You will have no gradient and transport will stop. What you got to do? You got to get rid of the intracellular sodium by pumping it out in the extracellular fluid. What about the concentration of sodium in extracellular fluid? Is it high? Oh yeah. It's like, what? I don't remember the rate. It's like 70 times higher than in the cell. It's huge. So sodium cannot diffuse in the extracellular fluid. You have to pump it. So this is the pump here. Those red arrows represent pumps. Now, one of the problems with explanation of physiological concepts that I personally have is that these processes are continuous. Okay? It's not like pump works, then stops, and waits for sodium to diffuse. Okay? It all happens at the same time. Pump, by pumping sodium out of the cell, constantly removes sodium and maintains its low concentration in the cell. Does it make sense? That allows sodium to diffuse from the lumen of intestine here into the cell. Does that make sense? And during that diffusion, you have a co-transport. This is called the amino acid sodium symporter or symport. Import, it doesn't really matter. Okay? Together, these two chemicals, sodium ion, diffuses, and energy of that diffusion right here fuels the uptake of amino acid. Does that make sense? Now, concentration of amino acid in the columnar cell is high. So it's readily going into the blood. Now this is diffusion. Nothing actually happens in the extracellular fluid with amino acids. I mean, they're not used for the protein synthesis, okay? The only reason why they're there, they get delivered to all other cells in the body. Okay? Does that make sense? So, this is the facilitated diffusion through the amino acid carrier protein. Does that make sense? No energy required. Now this is secondary active transport. When it's leaving the lumen, it goes together with sodium by secondary active transport. But once it's in the epithelial cell, it can leave epithelial cell through the basal membrane, okay, basal side of the cell, just by facilitated diffusion. No energy is required. Does that make sense? Now, active transport. When we say active transport, we usually suppose energy is needed. Do you see any ATPs that on the left slide here? You see any ATPs? I don't really see any ATPs. But energy means ATP. Where do we spend ATP? What actually uses ATP? Something on that slide uses ATP. It's indirect thing. It's not like written out. I use ATP. Not the Symporter does not use ATP. It's because of the sodium diffusion amino acid can go. It's not Symporter. Some other transport molecule. It's not on the left side. Sodium pump. This pump uses ATP. So you see, it is sort of an indirect use of energy. Pump uses ATP, creates the sodium gradient, sodium diffuses, okay? Its diffusion provides energy for amino acid uptake. Does that make sense? So absorption actually requires a lot of energy, as you can imagine. So we spend a lot of energy to get some energy. Okay? Yes. 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 Absorption is entirely intestinal function. Moreover, absorption of nutrients is entirely function of small intestine. There's virtually no nutrient absorption in the large one. Okay? Does that make sense? Good. Now, is that clear? I will ask you about transport on apical membrane. Okay, transport on basal membrane, whether it's primary, secondary active transport, facilitated diffusions, and so forth. Okay. Yes. Oh, sure. Okay, step by step. If you ingest pathogenic bacteria, now your question is, will bacteria make it into the intestinal cells?
It's like an intestinal bug. When does it activate?
When it causes symptoms, when you start to shit yourself. Depends on the bug. Some of them will produce the toxin. Like Shigella will produce the toxin, Kohler will produce the toxin. C. diff will produce the toxin. And that toxin is what's causing the symptoms. And it can be different. Like cholera blocks the chloride uptake, cholera toxin. So chloride stays in the lumen, water stays in the lumen, and you have 20 diarrheal bouts a day or something like that, more than 20. Okay. Clostridium difficile produces cytotoxin. It destroys cells, inflammation, enterocolitis. Okay. Worked, but symptoms are going to be similar because if columnar cells are dead, they cannot do absorption. Everything stays in the lumen, including water, and it comes out. Okay, sometimes with the blood. Some of those guys will make their way. Bacteria may not make their way into the epithelium. Well, some of them will. Some strains of E. coli will. Some of them will make their way onto the epithelium. Does that make sense? Because interesting fact, you all know that we have a lot of microbes in the guts. There is a gap between epithelial cells and microbes. Seriously, they don't attach to the cells. For some reason, in a healthy intestines, there is a gap. Like, this is where epithelial cell ends, right? And this is like where bacteria start. This is free space, no one's land. Okay, we don't really know why. And we know that if there's no gap, there's a disease. Okay? I'm going to go for carbohydrates, and it's extremely simple because it's the same. Seriously, it's the same. Okay? So remember, we start with a starch like this. Starch consists of many, many, many glucose molecules. Okay? Now, amylase breaks it down to short oligosaccharides, right? Like maltose or maltriose or different sorts of dextran. And then dextrase or maltase will break it down to glucose. The enzymes of the brush border. Does that make sense? Remember, big, smaller, smallest. So the smallest guys, you can see them over here, okay, will be taken into the columnar cells. Again, for glucose and galactose, the secondary active transport will be used. Okay? Fructose, since its concentration in the columnar cell is negligible, we don't make any fructose, okay? It will pretty much diffuse into the columnar cell and then out of the columnar cell into the blood. Does that make sense? So you have secondary active transport here, facilitated diffusion for fructose. Glucose and galactose use secondary active transport. Again, because of the sodium. So sodium diffuses into the cell, takes with it carbohydrates, okay? Takes carbohydrates. Carbohydrates then use facilitated diffusion to enter the blood. All right? And here's the nice little illustration of sodium maintaining, sodium-potassium pump maintaining low sodium concentration. What I pretty much figured out here. Okay. So it's the mechanism, the basic mechanism is the same as for amino acids. Secondary active transport on the apical side. Facilitated diffusion on the basal side. Okay? I'm going to stop now. I'm not going to go into the fats because it's like another 15 minutes of just talking about this. But so far, we've covered amino acids. We've covered carbohydrates. I want you to know the idea of big, smaller, smallest. Now, if I ask you the question, please describe the mechanism of carbohydrates, digestion, and absorption. Please don't tell me big, smallest, smaller, smallest. It's not sufficient, okay? Tell me large polysaccharide molecules are broken or hydrolyzed into smaller pieces by pancreatic amylase, okay? And those smaller pieces, such as maltose, sugar, or maltriose, or dextrose, are further digested by the dextrase and maltase of the brush border. Okay, two individual monosaccharides taken into the columnar epithelial cells, primarily via the secondary active transport, and blah blah blah. Okay, that makes sense? Okay.